So today we'll show you how to do streaking. So you have a loop, just heat it. So it should be red hot. Okay. So once it cools a little bit, then you have to put it somewhere on the plate to cool it down. Okay. Okay. So then this is your YPD, right? So this is your plate. We want to take these yeast colonies. So whichever is the good. So near the heat, we'll lift this up, touch one colony like this, okay, and then open it and do the smooth movements like this. So hopefully you'll have a colony tomorrow. And then you, so always write on the back side of the plate, not on the front side, because you can change the lid, okay, but the media is attached to the lower end, so you should always do. So this is YPD SH1, okay. So this is year 1. Then we'll take another colony, again heat it red hot. Okay, so you have to burn all the previous bacteria or residual material that's attached to the loop, then kind of cool it, okay, on one side. So once you cool it, you can take a colony on this plate. And then slight moments, and this is called streaking. Okay, दो है ना. अब हो गया तुम्हारा ये येलो वाला लेना है, ठीक है. So with slight hands, you'll have to move it like a feather. Okay, this is also your YPD. We'll call it SH3. This is SH2. YPD. Okay, this is YPD. And today is 8th. Huh? 8th.
Yes, sir. 8/7.
So these will be your yeast cultures. So this plate we have used, you will not throw it, you will keep it for a while. This plate we have used, right? इसमें -1 है ना?
जी सर.
Okay. One more we'll take. So heat it, okay. Cool it at the end of the plate, okay, so that you don't burn the bacteria, the fungus or the yeast. So So we'll take this colony, just touch it like this. Okay. यहाँ हम स्ट्रोक्स ज़्यादा देंगे, ठीक है, फिर आहिस्ता आहिस्ता उतरेंगे नीचे, आहिस्ता आहिस्ता। सॉरी, ठीक है। ये हो गए 4 तो तुम्हारी काफ़ी, हम संभाल के ये नहीं फेंकोगे अभी। YPB SH487 ये हो गया आपका क्या, यीस्ट। अब बैक्टीरिया आपका एक ही आया है, तो हम एक ही कॉलोनी को इसी लेंगे, या 2 इसमें, येलो भी है और ये भी है, ठीक है, हम 2 लेंगे। तो इसके लिए हम LB लेंगे। LB, so here you have to be careful. You have to, there are 2 types of colonies, okay? One is your yellow one is your orange one. So we'll have to take both, but we'll be careful. I'll select a red colony that is alone or aloof somewhere. Maybe this. This is LB SH5, okay, 87, and we took one. Now we'll take one yellow colony. कई बार से खुद करना है, ठीक है। Heat it till it's red. है तास्मीन, now this colony you see here is alone, right? So we have to be careful not to touch anything else. So you'll always sterilize the loop once you're done. And this is your sample number 6, huh? 6, LB. Okay, so you'll incubate them at 37 overnight. So tomorrow morning you check your cultures.